For most of human history, aging was assumed to be inevitable and unstoppable. That assumption is being dismantled in real time by longevity science. Research has now identified specific molecular mechanisms of aging, and specific compounds that measurably slow or partially reverse those mechanisms in animal models and increasingly in human trials. This is not anti-aging marketing. This is peer-reviewed biology appearing in Cell, Nature, and Science. In 2023, a landmark review in the journal Cell updated the hallmarks of aging, the biological mechanisms responsible for the decline that comes with time. There are now 12 recognized hallmarks, including genomic instability, telomere shortening, epigenetic alterations, and cellular senescence. What is remarkable is that researchers have identified specific molecules that address each of these hallmarks. Not hypothetically, in living organisms. Animals given these compounds live longer, function better, and show measurable reversal of aging biology. The research for this video is drawn from the field's most authoritative sources. The 2023 Cell paper by Lopez-Oding and colleagues updated the biological hallmarks of aging framework. Research in cell metabolism by Mills and colleagues documented NMN effects on aging in animal models. Work by Blagascloni in the journal Aging examined rapamycin's role in lifespan extension. And David Sinclair's research at Harvard has contributed significantly to understanding resveratrol and sirtuin pathways. The most fundamental misconception about aging is that it is a random, uncontrollable entropy, like a car simply wearing out. Longevity science reveals that aging is a regulated biological program driven by specific molecular processes. The hallmarks of aging framework identifies 12 distinct mechanisms, each with specific molecular players. This is important because you cannot target something you cannot define. The scale of the aging biology problem is significant. Research shows that biological aging, measured by epigenetic clocks, can diverge substantially from chronological age based on lifestyle factors. People with the same birth year can have biological ages differing by 10 to 15 years. The hallmarks of aging, senescent cell accumulation, declining NAD+ levels, mTOR pathway overactivation, telomere shortening, accelerate in people with poor lifestyle habits. They are also slowed measurably by specific compounds and practices. Research comparing fast agers versus slow agers at the molecular level reveals clear biological differences. Fast biological agers show accelerated accumulation of senescent cells, zombie cells that cannot replicate but release inflammatory signals damaging nearby tissue. They show faster NAD+ decline, limiting cellular energy and DNA repair. Their mTOR pathway stays chronically activated, preventing cellular autophagy and renewal. NMN, nicotinamide mononucleotide, is one of the most extensively studied anti-aging compounds in the current literature. It is a precursor to NAD+, nicotinamide adenine dinucleotide, which is required for hundreds of metabolic reactions and for the activity of sirtuins, proteins that regulate DNA repair and cellular health. NAD+ declines by approximately 50% between ages 40 and 60. The second compound with exceptional evidence is rapamycin, an mTOR inhibitor originally developed as an immunosuppressant. Research has shown that rapamycin consistently extends lifespan in animal models, including in mice where treatment began late in life. The mTOR pathway acts as a cellular growth accelerator, useful when young, but associated with accelerated aging when chronically overactivated. Rapamycin dampens this pathway, enabling more autophagy and cellular renewal. 3 additional compounds round out the current top longevity evidence list. Resveratrol, a polyphenol in red grapes, activates sirtuin proteins that regulate DNA repair and stress resistance, though human trial results are mixed on bioavailability at practical doses. Metformin, a diabetes drug, activates AMPK, which mimics some effects of caloric restriction. Several large aging prevention trials are underway. Each of the 5 compounds targets distinct aging hallmarks, and the mechanistic logic behind each is well established. This is not pseudoscience. These are pathways under active investigation at Harvard, Stanford, Mayo Clinic, and dozens of leading longevity research institutions. The hallmark each compound targets, its mechanism, and its current evidence level differ, but all 5 have demonstrated measurable anti-aging effects in research settings. Human clinical trials for NMN, Physodin, and metformin are already producing published data. The longevity science framework suggests thinking about anti-aging compounds in 2 categories. The first category is lifestyle compounds, those achievable through diet and behavior. Resveratrol from grapes and berries, quercetin, and physodin from fruits, NAD+ precursors from niacin-rich foods, and caloric modulation that activates AMPK naturally. The second category is supplemental compounds, those typically requiring supplementation for effective dosing, NMN, NR, and potentially metformin. The research suggests that lifestyle comes first. Compounds amplify what lifestyle already does. Longevity research has documented measurable biological outcomes at different time points for these compounds. NAD+ restoration with NMN or NR produces measurable serum NAD+ increases within 2 to 4 weeks of supplementation, confirmed in human studies. Senolytic clearing of senescent cells with Physidin, studied in high-dose intermittent protocols, shows measurable reductions in inflammatory markers within weeks. Epigenetic age reversal, documented with combination protocols including rapamycin, requires months but has been measured using validated biological aging clocks. Research supports a practical longevity compound approach accessible to most people. Start with dietary compounds. Eat blueberries, strawberries, red grapes, and onions regularly. Each provides Physidin, quercetin, or resveratrol. Practice time-restricted eating for 16 to 18 hours to activate AMPK and autophagy. If interested in supplementation, NMN or NR at 250 to 500 mg daily has the most current human trial data. Consider resveratrol at 500 mg with a fat-containing meal for better absorption. The research logic for longevity compounds is layered. If you eat a polyphenol-rich diet and practice time-restricted eating, then you are already activating AMPK, sirtuins, and autophagy, the same pathways targeted by pharmacological compounds. If you add NMN or NR supplementation, then you address the NAD+ decline that cannot be fully reversed through diet alone. If you are interested in Physitin senolytic activity, then high-dose intermittent protocols, studied in the Mayo Clinic research, show the most documented. Longevity science has identified 12 hallmarks of aging, and 5 compounds with the strongest evidence for addressing them.
NMN restores NAT.
Rapamycin inhibits mTOR. Metformin activates AMPK. Resveratrol activates sirtuins. Physitin clears senescent cells. These are not theories. They are mechanisms demonstrated in peer-reviewed research at the world's leading institutions. The practical approach begins with diet and fasting, which activate the same pathways naturally. Supplementation then targets what lifestyle alone cannot fully address. The science of longevity is no longer speculative. It is mechanistic, targeted, and advancing rapidly. Start with what the research supports at the dietary level: polyphenol-rich foods, time-restricted eating, quality sleep, and regular exercise. These activate the same molecular pathways as the pharmacological compounds being studied. If supplementation interests you, NMN or NR has the most human trial evidence currently. And follow this research, because the field is moving fast, and the findings are genuinely remarkable.